Okay. I have for like the fourth time tried to update the link to the YouTube channel on Canvas. If you guys get a moment today, check and see if it actually takes you someplace now, and we'll see how it's going. I tried to upload the last couple of videos too. Okay. So we left off on Wednesday talking about motor neurons and where motor neurons are located in the spinal cord, right? And so each individual muscle has a pool of motor neurons, hundreds or thousands of them. And as you can see here, we've basically zoomed in on a couple of vertebrae, right? Sort of lower lumbar vertebrae, high sacral vertebrae, right? And you can see where these lower body motor neurons are going to exist in the spinal cord. Okay. All motor neurons that go to the same muscle are in roughly the same location in your spinal cord. Okay, so we visualize this kind of as like these tubes or whatever of motor neurons that are going to exist right there. All right, so here's a fun question. Why have them all in the same place? What's an advantage of all of the motor neurons that go to your quads being in basically the same place in your spinal cord, as opposed to having them sprinkled all up and down the entirety of your spine? Hi, what do we do when nobody answers?
Squats.
Okay. Do we need to squat? Should we all stand up? You want to make use of your quads and stand up? Everybody stand up. Come on. Do this. Okay. It's not going to be. One thing you learn as a parent is you cannot make idle threats. Okay. So let's use our quads. So squat. Let's go. Squat. It's Friday morning. Make some legs. Let's go down and hold it. Just down and hold. Hold. Okay. Now, while you're holding, stay. We're going. While we're doing this, okay? Why is it a benefit to what you're doing right now that all of the motor neurons to your quads are in the same place? Emily, I see you. I see those of y'all that are sort of helping ourselves. Very genius. Imogen.
Yes.
It's more efficient how? Why? But they're all in the same. So if they were different, you're close. What's happening? Go quickly. Right? Okay, Trinity. It's easier to recruit. We're close. Yes. Imogen, you're really close. All right, y'all stand up. Okay. Okay. If I had some quad motor neurons in my cervical vertebrae, some in my thoracic vertebrae, and then the rest down where they normally are, right down in my lumbar vertebrae, which ones, when the brain sends a signal, are going to fire first? Cervical. Okay. And then which ones are going to come next? Thoracic, and then the rest of them. What if, okay, y'all can sit down, okay. What if I need to generate a huge amount of force? I need all of them activated right now. What happens if cervical go first and then the next ones and then the next one's down? Am I going to get all of them on at the same time? No. Okay. Am I then going to be able to, as fast as possible, get as much force as possible? No. Okay, and so that's the advantage of all of them being in the same place. Okay, of all of them being next. Yes, we can send one signal from the brain, which is going to freak you out. We'll talk about that later today. Okay, and we'll get them all on, we'll get some of them on. But you get a much more coordinated, smooth movement because they're all in the same location. Okay, and it's not like, oh, there's some here, and it's kind of herky-jerky as it slides down. Let me get the next group and the next group and the next group. So that's the benefit of all of them being in the same place. What's the downside? They're all in the same place. Okay, stand back up. Okay, wait, we gotta take her, Justin.
Get injured in that spot.
That's right, okay? When you damage the spinal cord, everything below the level of injury doesn't work. Okay? So if you had them spread out, you might still have some function in the vast majority of all of your muscles, but your force would be less, but you might still have some function. Okay? The way that it is though, because they're all in the same spot, you basically either have all of them or you have none of them. Some of them, they're gonna cross vertebrae, so you know, you might get a little bit, but that's the downside. Okay, that's the downside. So if something happens and it's either a spinal cord injury or it's something on, right, so if you're gonna have something where the nerve bundle leaves out of the foraminal space or something, you kind of lose all of it, right? So there's good and bad to all of this. And that's kind of what we need to understand about where they're located and how all of that stuff is gonna work together, okay? Okay. So again, we'll mention briefly a couple of things here, and we'll come back to some of this a little bit later on in class. What you see here is a vertebrae, okay? And you can see basically on the top is the dorsal side, on the bottom is the ventral side. So the top part here is what faces out in your back, okay? The ventral side is what faces in towards your internal organs, okay? Your motor neurons exist, all of them together in the ventral side. They're all in that, where you see that red and yellow kind of coming in there, right? That's where all the motor neurons are, okay? At that same level in the spinal cord though, you're going to have what we call afferent neurons that are going to gather information from that same muscle or that same location in your body, and that information is going to come back in and feed in on the dorsal side. And it's really important that that information is going to be together, okay? Because as we'll see in a week or so, information coming back from your muscle, where you are in space, how much force it's producing, what is the length of that muscle, is going to directly provide signals over and across that vertebrae to those motor neurons. And having those in close proximity is really important. Okay? So that's where they're located. They're all together, but they're on that ventral side. And then you see where all of those axons are going to bundle together, and they're going to exit together in what we call that spinal nerve or whatever, which then becomes what you think of and you see when it wraps in together with the afferent nerves as sort of those large nerve bundles that we really call nerves. Okay? And so damage in the cord, damage in spaces where it leaves out, that's called the foraminal space on either side, can cause some real serious motor impairments. Okay? And so, but you can trace then, my bicep doesn't work, I don't have feeling in my bicep, but I do in my tricep. You can trace all of that stuff then back potentially to either that part of the periphery, or you can trace it back to that spot in the spinal cord, or that spot in the brain areas that control those. Okay, this is what you do a lot of if you're a physical therapist or an occupational therapist or a PA or an orthopedic surgeon. Is somebody shows up and says, I can't move my arm, right? Or I have numbness in this place, but not in this other place. And you have to work your way backwards from form and function to try to help you eliminate and diagnose what's happening. Okay, so that's part of why I think understanding all of this is so important, is that it all fits together in a way and helps us have proper function, but you can see improper function and use that to diagnose things that might be going wrong. Okay, we talked about the motor axon the other day. Okay, and so I do want you to know a little bit about the axon. I want you to know about myelin. I want you to know about the things called the nodes that run VA, which are the gaps in the myelin along the axon where all the sodium and potassium channels are that allow the action potential, in essence, to jump its way down the axon and help us go faster. Okay, what we're going to spend a little bit of time before we do our activity data talking about is the neuromuscular junction. Okay, neuromuscular junction. This is the place or the location where the axon runs all the way down. And it branches off when it gets down near an individual muscle fiber. And the junction is where that nerve is going to be in very close contact. It does not touch, which is an important thing, but it gets in very close contact with individual muscle fibers. Okay? And that's how we transfer the signal from the brain, down the spinal cord, to the motor neuron, down the axon. Now we're going to jump it onto a fiber. So we can tell that fiber activates your cross bridges, generate force. Okay, so we're going to jump into a little bit of stuff about the neuromuscular junction. There are some intervening slides about myelin and the nodes of Ranvier. Look those over. It's all stuff you guys should have seen before. Okay, this is a cartoon of a neuromuscular junction. One of my favorite structures because I'm super nerdy in the midst of this, but if you look in the top left, you see, right, there's the motor neuron, it's in the spinal cord. There you have the axon, right, with this myelin on it that's going to come out. So lead all the way down, way down, out to an individual muscle fiber. Okay, so you see the fiber, and then you see kind of what it looks like where the end of the axon branches. Okay, we'll talk about why that branching is important in a second. It branches, and then you get these little nodes or nodules where it bulges out, which is also really important, that comes down and sits in very close contact with the surface of that muscle fiber. Okay. So what you see on the right, we've blown up basically one junction, one of those little areas. All right. And there's some structural things that happen here that are really important. Okay. On the end of the axon, it branches. That gives us more surface area. You can have more places of close contact. All right. But then at the end of each of those branches, it bulges out a little bit, it kind of balloons out a little. Okay. We call that, and I'm not going to ask you this, but we call this end of the axon you see on the right. That's called the bouton, B-O-U-T-O-N, okay? But it's going to bulge out, which also increases surface area, which is really important, okay? It also, by having more surface area, is able to hold more of the neurotransmitter that we're going to release into the junction to jump our signal. That neurotransmitter is acetylcholine, okay? Is acetylcholine. You can see in those little light blue circles on the right there, those are vesicles, and they contain acetylcholine. All right. On the bottom, where you see those folds, that's the cell membrane of the muscle fiber. That's called the sarcolemma. Okay. Under the bouton, sarcolemma folds. Okay. It functions much like the folds in your small intestine. Why is your small intestine folded? Do you know? Can you guys imagine you've got like 20 feet of small intestine all wound up in here? Yes, Kate. Increases surface area. Very good. Okay. Why is having more surface area good? Why would you want more surface? Improving the small intestine improves your ability to absorb nutrients. Very good, Justin. I saw a hand right there on the junction side. Why is it good? We're not absorbing nutrients. What are we trying to, maybe not absorb, but what are we trying to send across? That's right. Very good. Okay. So again, here is anatomy imparting function. It costs energy, okay, to make acetylcholine, to make it in the motor neuron and move that shit down like feet of axon, okay, to get it to the neuromuscular junction. That costs us energy. Okay. So when we release it, we want to make sure, one, that we really need to release it, and two, that it's going to actually go where we want it to go. So we want to release the minimum amount of acetylcholine to get the maximum effect. And one of the ways that we do that is, right, we have the folds in the sarcolemma. And in those folds are going to be receptors for acetylcholine. And this way, by having more surface area, we can have more receptors. Okay. So for every little bit of acetylcholine that gets released from the nerve, we have a greater chance of capturing that on the sarcolemma and transmitting our signal across. Okay. Does that make sense, guys? Do y'all kind of understand sort of how and why that's going to be the case? Okay. Okay. So we call the area on the top, kind of the bigger area between the end of the bouton and the sarcolemma is called the primary neuromuscular cleft. And then the area down in those folds is called the secondary neuromuscular cleft. And the function of those clefts is just to house acetylcholine receptors. And so this way, when we get a little bit of release, we're going to grab it, and then we're going to initiate an active potential on the muscle fiber, on the sarcolemma. Okay. As soon as we release acetylcholine, though, there is something that is housed in a thing called the basement membrane on the sarcolemma, the stuff that's kind of right along the outside, that's going to try to break the acetylcholine back off of its receptor. Anybody remember what that is from human phys? Health and fitness. I was bragging on y'all last week to Dr. Pinku and to Chris and to Alex and Caleb. They've done such a great job. Cooper, what do you think? Remember what it is? It's acetylcholinesterase. There's an enzyme that sits out here near the acetylcholine receptors. And basically, imagine, okay, turn to your neighbor and shake their hand. Okay, turn to your neighbor, shake their hand. Possible. No. Arden's like, no, I'm not shaking hands. In the morning. Shake their hand, then karate chop it out. Okay. Right? Shake, break. That's what happens. Think of shaking hands as acetylcholine binds to its receptor, and then basically almost immediately someone comes in and tries to chop your hands apart. Okay. That's what's happening here. So we make this connection. And then we break it off immediately, because when you make the connection, that's one of the steps that leads to force being generated. Okay. And then we want to break it off so that we can turn that force off really quickly if we need to, or if we want to. Okay. So that's kind of how all of this is going to go. Okay. So this shows a little bit more information about those things. It's going to talk about that we need 2 acetylcholine to bond. All right. So that's going to be a big deal there. All right. The next piece before we work on kind of our signaling side of things is we talked about this on Wednesday, but it's the idea of something called a motor unit. Okay. A motor unit is one motor neuron and all of the fibers to which it connects. One motor neuron and all of the fibers to which it connects. That's going to be on your test. Okay. I mentioned the other day that motor neurons come in different sizes. Okay. So as you can see on the left, you've got a motor neuron that is 6 fibers. Okay. And then on the right, you've got one that I don't know what that is. Is that 10 or 15 or maybe 20 fibers? Okay. You have small motor neurons, medium-sized motor neurons, and then very, very large. They can range from 10 to 20 fibers into the tens of thousands of fibers that connect to one motor neuron. Okay. The motor unit is the functional unit, right, by which the nervous system turns on skeletal muscle to generate force. Okay. If I told you guys activate one motor unit's worth of force in your finger, think you could do that? You can do one motor unit? No? You do it, but you'll never think about it now. You're not like, okay, squat, okay, I need 7 motor units, and now I need 8 because it's getting kind of hard. And now I've done like 14 reps. I got to get a 12th one in there, right? We don't think about it that way, but that is exactly how everything progresses. Okay. I go to try to pick something up, and I start off with 1 motor unit. That's not enough force, so you almost immediately 2, 3, 4, 20, all the way up. we adjust our force production from the nervous system side by adjusting how many motor units we recruit. Okay? Small motor units connect to muscle fibers that are slow twitch. Okay? This is absolutely important. Okay? Small motor units are made up of slow twitch fibers. Okay? The largest motor units are made up of the fastest fiber type. So they're usually type 2X. Okay? So when you start recruiting or activating these motor units, you're always going to activate the slow twitch ones first. And then progress your way up to the 2X. Okay? If it was the opposite of this, then every one of you that's trying to write stuff or type stuff or anything else right now, you'd already be fatigued. Okay? You try to write and you might make like 3 letters and your 2X fibers would be fatigued, and it wouldn't work. Okay? The other thing that this lets us do, one, it helps us prevent fatigue during low force contractions. But the other thing that it does is by starting with the small ones, is it allows us to have fine motor control. Okay? Fine motor control. Anybody play an instrument? Anybody play the guitar or the violin or any kind of thing like that? Or you play the piano or something, right? Lots of studies on sort of expert musicians. They have the best fine motor control, like of anybody that we can find. Okay? And what that means is you can very precisely alter and change very, very low force contractions. That's fine motor control. You don't need much force to be able to write, okay? But it takes very small, very precise changes. And so by turning on the small ones first, it lets us gradate our force production in very, very small increments. If you activate a motor unit first, it has like 10,000 fibers in it, you're going to get quite a bit of force. Way more force than a motor unit that has 10. Okay? And so if you go from 10, and then the next one has 15, and the next one has 20, right? We've still only activated about 40 fibers, as opposed to those 2X ones that might have tens of thousands of fibers. So this pattern of small ones first, slow ones first, we prevent fatigue and we have fine motor control. Okay? It's vitally important to our way of life. If it was not set up this way, we couldn't write, we couldn't do things in the way that we do. Okay? So that's the way this setup is. All right. So I will briefly mention an action potential, and then we're going to jump down and we're going to talk about excitation, contraction, coupling, and I'm going to have you guys do a thing, a little activity. Okay. You guys have learned about action potentials before, yes? An action potential is electricity. That's what it is. Okay. It's a small, very, very brief zap of electricity that we generate in the nervous system or in other excitable tissues, and it moves. Okay. We use electricity as a signal. We use electricity as a signal because it's precise and it travels really fucking fast. Okay? Touch the hot stove, pull it away. Okay? Because action potentials can move through the nervous system in, microseconds. Very, very, very short amounts of time. Okay? So we need a couple of things about nervous system cells and about your muscle cells in order for them to generate action. This is the case for all what we call excitable cells. Okay, if you can generate an action potential, it's called an excitable cell or an excitable membrane. You need what's called a resting membrane potential. And a membrane potential means we need a difference in positive and negative charge from one side of the membrane to the other. And then we also need concentration gradients of different ions, of positive and negative ions across that membrane. Okay? And we generate action potentials by having these differences, and then we open up a channel or a doorway, right? across that membrane, and the ions move down their concentration gradients, yay chemistry, okay? And as the ions move, the charges go with them. So that difference in charge from inside to outside will flip, and that generates electricity, okay? When you generate static, right, you rub things together, you're just accumulating charges, and then you touch somebody else, or you touch something, and that charge moves. That's how lightning is created, right? We accumulate these charges, and then at some point, those charges flip, and they shoot down to the other place. And that's how we get an action. Okay? I've got like 10 slides trying to show you guys how we generate action. All right, but the big thing that we need, and the way that this is going to work, is we have this resting membrane potential. Okay, and so what you see here is outside the cell on the top, inside the cell on the bottom. And in general, in nervous tissue and in your muscle fibers, we're more negative inside a cell than outside. So you've got about minus 70 millivolts of negative charge on the inside of the cell. And so then when we open channels across those membranes, then positive is going to want to move from outside to inside. We go from being negative to positive, and that generates our action potential. Okay. The two big ions you need to know that help us generate action potentials are sodium and potassium. Okay. Sodium and potassium. We have lots of sodium outside of our nerve cells and outside of our muscle cells. And you have way more potassium inside. Okay. And so those two things are going to move when we open our channel. Sodium comes in, it's positive, so it makes the inside of our cell more positive. It goes from negative to positive, boom, action. We then are going to take that potassium and it's going to leave the cell. And as it leaves, it is going to take that positive charge that we generated from all the sodium coming in. And as it leaves, it becomes less positive and we get our negative charge. Okay? That's how we're going to move these two sorts of things. But now we have a problem. We've done an action potential, we've repolarized, but now we have a bunch of sodium inside when it needs to be outside, and a bunch of potassium outside when it needs to be inside. So we have what are called ion pumps that use energy, and they close the channels, and they pump the sodium back out, and they pump the potassium back. When you guys aren't exercising, there are estimates that somewhere between 25% to about 35% of all the energy that you expend at rest is from running the sodium potassium pump. Okay, it's simply repolarizing neurons and things like that. It's why. Okay, so very, very important. All of this happens in milliseconds. They move, you get an action potential, then we immediately repolarize. It happens really, really fast. So you can then get another action potential and another and another and another. Okay. Okay. I'm sure in human phys that you guys had to learn all of this, right? This is the steps in the process of making an action potential. What I want to show you guys basically is how this works at the nerve level. Okay. You guys remember we have dendrites that are going to gather signals to that motor neuron. Okay. As those dendrites begin to pick up signals, they may generate what's called a gradient potential. It's like, here's a little bit of a signal. It's your neighbor tapping, right? It's like, hey, 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 hey. And at some point, you're like, okay, that's enough, right? Stop. Okay. Gradient potentials are that initial, hey, hey, hey. You guys may not have that experience. When you have a 6-year-old at home, you get a lot of dad. Like, what? Okay, on those kinds of things. That's a gradient potential. You're moving that cell. Those signals are moving it towards generating an action potential. It moves it from -70 up towards its threshold potential. Once you get a big enough gradient potential, then it opens the sodium channels. Sodium rushes in. We get this spike. You depolarize. action potential. Then we're gonna repolarize by closing the sodium channels, opening the potassium channels, comes back down. It overshoots a little bit, that's called hyperpolarization, and then we restore our ion. Okay. Relatively straightforward, relatively simple stuff that's happening here. Okay. If you really care, I'm not gonna ask you to do this. This is I think a nice visual showing the action potential in red, and then when sodium moves and when potassium is moving in those purple things, so you can see that those are not gonna exactly line up with each other, but you can see how we get depolarization from sodium, and then the action potential turns itself off as the sodium stops moving and the potassium does. And then I got a bunch of things trying to show you guys, sort of what all is happening with all of this. Okay. All right. I want to do one last thing, and then we're gonna do kind of a big, all-encompassing think project. You get an action potential on the neuron. It goes down the axon. It reaches, moves on. You release acetylcholine. Acetylcholine binds the acetylcholine receptors, and that opens sodium channels on sarcolemma. So you go after acetylcholine, then that creates an action potential in the muscle fiber. Once that happens, it travels along the surface of the muscle fiber until it reaches a T-tubule. And then it goes down that T-tubule, down into that tunnel, if you will, and reaches those receptors that we mentioned like a week ago. It reaches the dihydropyridine receptor, which then signals to the ranitidine receptor, and the sarcoplasmic reticulum releases calcium. Okay? This process, where the sarcolemma depolarizes, the T-tubule depolarizes, and then we get calcium release, is collectively referred to as excitation, contraction, coupling. Okay? Excitation or action potential on the sarcolemma and T-tubule, to calcium release, which is the thing that initiates, right? Calcium binds to troponin, moves tropomyosin, we get a cross bridge, then we get a collagen. So it's an action potential leading to cross bridge formation. That's excitation, contraction, coupling. Okay. This is the process that gets messed up. Well, you guys were not able, and I saw y'all cheating when you were doing your squats. What was happening was this wasn't working correctly, and you weren't able to generate as much force. Okay, we're going to go with that enough that y'all were just cheating in the midst of everything. Okay, so what this is going to show basically on the left is acetylcholine binds, right? Sodium channels open, we get an action potential along the sarcolemma. It then comes down, right? And from here, it then travels along the sarcolemma, down the T-tubule that you see here in yellow, reaches DHP, that signals ranitidine, and calcium gets released. Okay? And then after that, we do the cross-bridge cycle. Okay? So that's how all of this works. Okay. We are going to, let me see what else that I've got. This is the same thing, and then we get the cross-bridge cycle. Okay. What we're going to do now with our last, I don't know, 12 or 14, 15 minutes of class is going to be this. Okay. I want you guys to get in groups, okay? And I want you to try as best you can to start in the brain, and I want you to make a list of all of the steps, all of the structures, all of the signals, where we're going to go from the brain, and we're going to signal all the way down until we initiate cross bridge and get the cross bridge cycle and get a power. Okay, all of that stuff together. Okay, I call this process cognition to contraction. It's from the brain thinking about I need to contract, and what are all of those steps that are going to come down to the brain. Okay, we will go through all of them next week, but I want you guys to try today. Ty and I will look at it and see how you're going to do. All right. So that's what we're going to do. We're going to turn that in at the end of class. All right. I'll try to wander around. Ty will wander around and see how you guys are doing. So Go. Where does it, what is it sitting there? Where does it go next? Okay. Awesome.
Yeah, right.
Where do you think we need to go next? Okay, what, how do you think the action? Hey, we spend 80 order to get back to Anderson. So the junction. Is someone riding? Oh, way down there. Yeah, how are y'all doing? Slowly but surely we're getting there, okay? Every structure, every signature, okay? Every little thing, every receptor, every aisle on the wall. Okay? And that would happen to recreate all of this on the chart, because we're starting. Okay.
Now, you're looking at
Yeah.
All right. We're out of time. We're out of time. So. Turn in what you have to Ty, please. If you wrote it out, make sure all your group names are on there. If you didn't write it out and you want to email it to me, that's great. Make sure all of your group members are on there, okay? Whatever you got done, you're getting credit for. We know it's not going to be perfect. You got to start somewhere with all of them, okay? All right. I'll see you guys on Monday. Have a good weekend. Make good choices.
Could I create like a little bit of a worksheet, maybe, and look over it that I could like post for them to use for this?